Thank you, Dr. De Robertis. Yes.
Yeah, thank you so much. A beautiful presentation. Actually, as we discussed, molecular drive is a kind of important issue. And it is amazing to insert CRISPR-Cas9 system by CRISPR-Cas9 to the genome. But right now, it is said that there are two major issues. One off-target issue, which means even though we have a guided RNA, the targeted site could be a different one from the original. The other issue would be a mosaic. Over the development, the cells, which are original one and the other one is inserted one, is a mosaic. So those two issues would be a very important problem right now for CRISPR-Cas9 and the related system. So in the case of molecular drive, and what do you think we should solve those kind of problem, or even though still to some extent we can go ahead to prevent kind of such as deleterious insects to prevent. So from your perspective, what do you think?
Well, I've just prepared this for this. It's not my field, but it will be very dangerous, of course. And then what becomes dangerous are off-target effects. And so some of the technologies that they use, they do all this, and then they target something, and then they inactivate Cas9 with another guide RNA against Cas9. Because if you have Cas9 active all the time, it can catch other RNAs and edit using the other RNA that recombined with the other part of the RNA. So there's a danger of having Cas9 in all cells, because it will start editing things that it shouldn't be editing. So that's dangerous. And I tell you, if we could eliminate Aedes aegypti, that would be wonderful. So if they could do it. So that would be one species that we should terminate. But it's a big problem, the mutation of the sites of this. But in the case of in the case of malaria, what they're doing, they're putting genes in this cassette, other genes that would target some other thing. So, all this, you see, both aspects is completely non-Mendelian genetics. So it's a big change.
In these diseases that are due to a mutation in one of the alleles, is it known whether the wild-type allele is expressed? That is one question. And the other question, do we know the molecular, the explanation of these diseases at a molecular level?
Yes. These are simple loss-of-function mutations. So if you have one gene mutated, the other one is working perfectly fine. But it's haploinsufficient, means you don't have enough of the dose of the protein. And so that's what's shocking, that we have so many haploinsufficient genes and we function just fine. But it's because of the dose of the gene that makes a difference.
So this may be a good time to, or there is one more, okay.
I wanted to ask a more general question. It looks like you have reached a stage in the ability to edit the genome where I am struck indeed that the very same mechanism that allows you to edit the genome can be inserted in the genome itself. So seen from the computer science point of view, I was wondering if this is a stage where you have something like a universal Turing machine capability inside the genome itself. And does that mean that there could be a sort of algorithm running on this genome mechanism? So is it too far-fetched to think about this sort of computation going on?
You know, the bacteria have it very controlled, and they use it for immunity for phage. So they register the last 10 phages that they've seen, so you can keep it under control. These things that we're now putting into a eukaryotic cell, they don't belong there, so I don't know if they can go out of control or not. But it has potential. I mean, it's not like it's just a Far West sort of thing. You know, many cancers, I'm changing, not you're answering your question, many cancers arise from translocations from one gene to the other. The translocation creates a new sequence. So that's a target for the gRNA right there. So you can do that. You can target HIV virus, which in principle, which in fact inserts itself into the DNA of only helper T cells, and then you could infect with adenovirus, the same thing, and only kill the cells that have the retrovirus inserted in them. So it has great therapeutical applications too. So I think not in the germline. It seems to me that anything that has to do with the germline is like should never be done ever. But
so then, thank you.